Welcome to Unit 6, Lecture 5. So Unit 6 I'm breaking down into a couple of different parts. So this is the first lecture of Part 2, the structure of DNA. Learning objectives. Identify the chemical components of DNA. Describe the clues that help scientists determine the structure of DNA. Explain what the double helix model shows about DNA. Nucleic acid structure is basically a line of repeating units, okay? And these repeating units are called nucleotides. Oops, that's written there. You read that. All right. So these repeating units, which if you remember from way back when, are called monomers, okay? And that monomer is basically, it's a nucleotide, right? They're the building block of both nucleic acids, DNA and RNA. And what happens is you end up getting them lined up in a row where they're joined together through this covalent bond here. So in this case, we've got one nucleotide, two nucleotides, three nucleotides, four nucleotides. Okay, and the nucleotide has three basic pieces. Right? A phosphate group, a sugar, and then a nitrogenous base. So there are the three different pieces to one nucleotide, which then a bunch of nucleotides come together to make DNA or RNA, which we'll talk about a little bit more later. But DNA and RNA are very similar molecules. DNA is made up of two strands. Okay, so we've got strand number 1 and strand number 2. Okay. So you can see this is the strand from the original slide. So we have the phosphate group, we have the sugar, and then we have the base, okay, which are then put together in a whole strand. So DNA is two of these strands put together, but what you notice here is the other strand, okay, is actually upside down. So if we were to think about the phosphate group being the top, all right. The one side is right side up, but the other side is actually upside down. And so what that's called anti-parallel. So that anti-parallel basically means that one strand is right side up, one strand is upside down. Now, scientists have more specific names for it because when we're talking about DNA on a very, very tiny scale that's all wrapped up, there's really no top or bottom. But for us in this 2D model, you can see top and bottom, or upside down and right side up. Okay? What we have also figured out is that there is what's known as Chargaff's rule. Chargaff is the guy who discovered it. But what it means is that the nitrogenous bases have set pairs. Okay? So A and T are always bonded together, and G and C are always bonded together. So on one strand of the DNA, I think it was A, G, C, T, okay? On one side of the DNA molecule, there was A, G, T, C. On the other side of the DNA molecule, there was A, G, C, T, okay? And that would be the same, that combination of A matching up with T, okay, and G matching up with C would be the same regardless of how many G's we have in a row or how many A's or whatever side of the molecule you're talking about. But it's really interesting to know that evolution, basically, has created this pattern. All right, because what it does is it facilitates some of the other things that we're going to be talking about during this section of Unit 6. In between, so once again, you can see here, nucleotide, okay? And in that nucleotide, you can see again those pairs, G to C, C to, okay? So we've got G to C here, we have C to G there, A to T, T to A. So you can see that, and once again, we've got one direction, the other direction. But in between all of that, okay, is what are these hydrogen bonds. So what they do is they're actually holding the two sides to each other. They're kind of like the glue. The hydrogen bond is kind of like the glue holding the two pieces of the zipper together. So if you think about DNA as a zipper, okay, those hydrogen bonds are those, the zipper being held together. Okay, and it's really important because what that does is that keeps its shape properly. Rosalind Franklin was one of the scientists that was really important for helping us understand the shape of DNA. So DNA is a helix, and she, using some old school photography, let's just say, she ended up taking a picture of DNA molecules and looking at them and realizing that this double, this helix shape with these nitrogenous bases pointed in towards each other was really important to help our understanding of that. And then Watson and Crick were able to use her her photography and notice that double helix, so that twisted ladder kind of look. So they were able to take her picture and put this whole structure together to be able to understand how it all works together and how it fits together. All right, I will talk to you later. Bye.